Immunomodulation by resin acids for periparturient dairy cows. During the periparturient period and early lactation, dairy cows are prone to low-grade systemic inflammation, which decreases their productive capacity. Here we aim to study the effects of coniferous resin acids on biomarkers of intestinal permeability and systemic inflammation in dairy cows during the first 10 weeks of lactation. We conducted a feeding trial with 36 Nordic red cows from 3 weeks prior to predicted parturition to 10 weeks postpartum. The cows were fed either a control diet or a tall fatty acid diet or a resin acid concentrate diet. Both of the resin acid containing diets provided 0.63 grams per cow per day of resin acids. The cows were sampled for blood from tail vein at weeks 2, 3, 6 and 10 postpartum, and the samples were measured for a variety of biomarkers of inflammation. The effect of time was statistically significant for all studied parameters. Here on the right-hand side you can see that the anti-inflammatory cytokine interleukin-10 was elevated by tall fatty acids, but not by the resin acid concentrate. The gastrointestinal barrier function biomarker intestinal fatty acid-binding protein tended to be decreased by resin acid concentrate, but was not affected by tall fatty acids. Here on the left-hand side, you can see that the cytokine interleukin-6 was elevated by tall fatty acids, but not affected by the resin acid concentrate. And on the right-hand side, you can see that the concentration of interleukin-8 was also elevated by tall fatty acids, but not affected by resin acid concentrate. In conclusion, the effect of time was statistically significant for all studied parameters, likely reflecting natural changes in systemic inflammation in dairy cows during the first weeks after calving. The tall fatty acid treatment resulted in mild immunomodulating effects, while the resin acid concentrate did not do that. On the other hand, the resin acid concentrate tended to reduce other infections and decrease the gut permeability biomarker in intestinal fatty acid-binding protein. The results suggest a different mechanism of action for resin acid concentrate and tall fatty acids. The potential benefits of these products on cow immune system during the periparturient period needs to be verified in future experiments. Thank you for listening.